Today our topic will mainly focus on molecular breeding techniques and their applications in crop improvement. In this lesson, mainly 3 sections will be included: introduction, transgenic breeding, and next generation breeding. Okay, now let me give you a brief introduction. The first question is, what is molecular breeding? Actually, the molecular breeding means molecular tools applied in the plant breeding. As we know, with the rapid development of molecular biology, molecular tools have greatly changed the traditional crop breeding. These techniques mainly involved in genetic transformation, gene cloning, marker-assisted selection, and next-generation sequencing. In fact, several transgenic crops have been widely planted, such as soybean. It is a herbicide-tolerant transgenic. Maize and cotton. Both of these 2 crops are insect-tolerant transgenic plants. In section 2, I will give you a detailed description of transgenic breeding. In order to get a transgenic crop, 4 steps must be included. First step is cloning of objective genes and constructing of crop expression vectors. The third step is genetic transformation, and the fourth is identification of transgenic plants. Next, I will give you detailed description step by step. The first step is cloning of objective genes. So we can use these genes used in the transgenic breeding. Actually, we have a lot of alternative ways to clone a target gene or objective genes, such as omics comparing, its transcriptome, genome, and proteome. Omics comparing can help us to identify genes of interest. Microarray technologies such as asymmetric gene chips have been extensively applied. And this is an asymmetric gene chip. In this small classic plate, it is a special mini glass plate. So in this chip, a large number of genes with lengths about 20 nucleotides are printed on a special mini glass plate. Each gene has a specific position. We can see every column of the gene sequence is printed in a small plate, and each gene has a specific position. When seeding libraries are hybrided with the gene chip, the expression profile of tens of thousands of genes can be simultaneously seen. By comparing their expression profiles, candidate genes can be easily identified. And also, RNA sequencing and eye track techniques can also be used in identifying two techniques are identification target genes at the transcription and the translation levels respectively. Certainly, tDNA insertion and the positional cloning techniques can also be used to identification of objective genes. However, these two methods are time-consuming. Next, let's see how PCR works in details. Actually, PCR machine is just like a DNA duplicator, and its temperature can change. When the temperature rises to near boiling, it's about 19 and 5 Celsius degree. The double-stranded DNA is separated. It's named denature. Then, when the temperature decreased, the primer sequence binded to the template DNA, and then at a slightly higher temperature, polymerase binded to the binded to the primer sequence and ended the nucleotide to extend the second strand. This is the first cycle of the PCR. After 30 cycles, as many as billion copies of a target sequence are produced from a single starting molecule. Okay, just now we showed the detailed working mechanism of a PCR machine. The second step of transgenic breeding is constructing of expression vectors. We know for every gene, 3 elements must be included. They are promoter region, terminator, and coding region. Promoters signals how much proteins to produce, and where and when to produce this protein. And the coding region encoded functional informations. For protein coding genes, the messenger RNA will direct the translation to a specific protein. For no coding genes, transcribed RNA will function in cells, such as microRNAs. So we can design genes expression by using these 3 distinctive elements of a gene. For example, if we use a constitutive promoter, such as 35S promoter, and drive a BT gene, the transgenic plant will express toxic proteins everywhere and anytime. So the stem borer and the leaf roller will be killed if they eat the transgenic plants. However, if we just use a leaf tissue specific promoter, the transgenic rice will not resist to the stem borer. In order to construct a proper expression vector, we need to introduce our genes, a target gene sequence, in a proper expression vector. Generally, there are 2 kinds of vectors must be constructed. The first kind is overexpression vectors, and knockout of repression vectors. This one is an overexpression vector. We inserted our gene in a normal direction and transcribed the normal message RNA, so the transgenic blends will overexpress a targeted gene or objective gene. But in anti-cell silencing technology, we must insert an objective gene in a reverse way. So the transcribed mRNA will complementary to the endogenous mRNA. This vector showed how to construct RNAi silencing vector. In this vector, the forward and the reverse element were included, and a spacer, commonly it's a natural intron segment, was inserted between these two segments. Recently developed CRISPR-Cas9 system is a very useful tool for site-specific genome editing. Okay, let's see how the CRISPR-Cas9 systems are working in details. Actually, CRISPR-Cas9 system is just like site-specific scissors. Cas9 protein just acts like a highly specific DNA scissors. It can specifically cut the DNA sequence. In order to knock out a gene, we need to introduce Cas9 protein and a guiding RNA constructs. In this guiding RNA, we must include a specific sequence. It's a target sequence. Cas9 binds to the guiding RNA and scan the cell's DNA, looking for word recognition sequences, two gene bases next to each other. When they find such sequence, Cas9 opens the DNA double helix to check whether the DNA and the RNA sequence is complementary. If they are not, Cas9 continues scanning. However, if they are matched, Cas9 will cut the double-stranded DNA. And subsequently, the cell can repair the damage. Just stick the two ends directly, and during this process, mutations often generated, such as the frame shift mutation. So, this kind of frame shift mutations will lead to the gene expression stops. And this allows scientists to use Cas9 to disrupt any genes of interest. And how to construct CRISPR-Cas9 knockout constructs. This slide shows the physical maps of constructs for CRISPR-Cas9 knockout It is different from the other kind of expression vectors. They use the U6 gene promoter or U3 gene promoter to drive guiding RNA. As we know, microRNAs are kinds of small RNAs which play important roles during the developmental regulation in plants. Which play important roles during the developmental regulation of plants. Recently, scientists have developed a new technique to block the small RNAs. It's named STTM. It's short tandem target mimic construct. And these STTM constructs including two short sequences mimicking small RNA target site and separated by a linker. It's about 48 nucleotide long. So each mimic sequence contains 3 additional nucleotides, so that a cleavage preventive bark could be formed. The expression of this STTM can lead to the degradation of the target small RNAs by small RNA degrading nuclease. The third step of transgenic breeding is genetic transformation. For crop transgenic research, 2 methods were commonly used. The first one is bacteria-mediated transformation, and the second method is microparticle bombardment transformation. In this method, objective genes need to insert in the teeding region, and in microparticle bombardment method, particles were coated with the DNA encoding objective genes. And after bombardment, the objective genes were integrated into the target genome. These 2 methods both of these 2 methods have advantage and disadvantages. The agrobacteria-mediated transformation is an convenient and precise method. However, it is limited for genotype and cannot freely applied in all kinds of tissues. Generally, tissues used for transformation is immature embryo, leaf discs, and apical meristem tissues. However, the particle bombardment method is more versatile, not limited by cell type, specific species, or genotypes. However, this method is expensive, such as the particles is made of gold. In order to identify transgenic plants, we can use all kinds of identification methods. At the DNA level, we can use a PCR detection, just like this picture shows, how to identify the transgenic soil bean. This is objective gene, herbicide-resistant gene, and the promoter, and the endogenous control, the negative control. And also we can use Southern blotting and the genomic DNA on PCR product sequencing to identify transgenic plant at the DNA levels, such as if we use the CRISPR-Cas9 systems to generate the transgenic plants, we have to use the sequencing method to identify, to check whether the targeted site is cut and mutations were generated or not. And secondly, we can identify transgenic plants at the RNA level. The methods including the real-time polymerase chain reaction and northern blotting and RNA sequencing techniques. At protein level, we can use proteomic comparing or Western blotting to analyze the transgenic plants. In transgenic plants, if the protein at the protein level, the transgenic plants have greatly changed. Also, we can use eye-track techniques to analyze the proteomic characteristics. Finally, we have to phenotyping the transgenic plants, such as the reported gene expression detection, the Gas gene, gray fluorescent protein, or red fluorescent protein. This is the reported gene expression detection. The most important phenotyping is to detect if or to observe the target traits. For example, if the transgenic rice plant harboring the Bt genes is resistant to the stem borer, and Bt cotton is resistant to the insect or not. And other traits they need to characterization, such as development and growth. And the backcross breeding is the final step of transgenic breeding, because most of plant species, only few lies or varieties will given high rates of transformation. And often they are lies with poor agronomic or quality characteristics. So transgenic plants are crossed with elite breeding lies or improved varieties, while traditional plant breeding methods, in order to combine the desired traits of elite lies and the transgene into a single lie. The offspring are repeatedly crossed back to the elite line to obtain a high yield transgenic lies, which express new traits encoded by the transgene. From 1996 on, insect-resistant Bt maize and cotton, as well as transgenic herbicide-tolerant soybeans and oilseed rape were planted on rapidly increasing areas. Globally, about 82% of total crop area for soybean, 68% for cotton, and 30% for maize were planted with GM, with genetically modified varieties, in 2014. In 2014, genetically modified crops were grown in 28 countries on total. In 2014, genetically modified crops were grown in 28 countries on a total surface of about 181 million hectares, which correspond to already 13% of the world's arable surface. farmers' profit increased by about 68% on average when using the transgenic crops, and also crop yield rose by 22%. The expense for pesticide declined by 39%. The last section is the next generation breeding. Using next generation sequencing technologies, we sequence an entire genome of transcription, become more efficiently and economic, and in great depths than never before. Here, we outline some important applications of next generation sequencing in crop genetics and breeding. Sequencing and resequencing, linkage analysis and association analysis, map-based cloning, and marker-assisted selection. Importantly, we can develop a large scale of molecular markers for linkage analysis, association mapping, and these techniques will greatly accelerate the development of crop genetics and breeding. This picture illustrated how next generation sequencing derived information can be used in crop breeding. High throughputing marker technologies coupled with advanced phenotyping platform provide new opportunities for discovering marker-trait association, which can sustain genomic-assisted selection and breeding. Genome sequencing information is enabling genome editing. It means site-specific mutagenesis. In next-generation breeding, there are two main strategies to assist breeding with molecular selection. The first one is MAPS. It means marker-assisted selection, which use molecular markers that map near or within specific loci with no phenotypic effects. The second is GS. It means genomic selection. Genome will exploit all available markers as predictors of breeding values. Actually, genomic selection has been applied to several traits in a variety of species, including maize, wheat, and rice. Overall, these studies suggest a wide application of genomic selection, even for species with large and complex genomes, such as bread wheat and maize. Okay, finally, let's have a brief summary. In this lesson, we discussed three sections: the introduction of transgenic breeding or molecular breeding. The second section is the details, steps, and the application of transgenic breeding. The last section is the next generation